Okay, this is the second session on cytotoxic chemotherapy, and in this case we're dealing with microtubule targeting drugs. And what we're going to do is look at what microtubules are and what are the main classes of drugs that target microtubules, how they work, and importantly what their resistance profiles are. So the three main cytoskeletal components that are particularly relevant to cell biology, these are actin, intermediate filaments and microtubules. We're going to be focusing on microtubules here. You just need to be aware of the difference between actin and intermediate filaments versus microtubules in terms of function. So intermediate filaments, which are not targeted by drugs, but it's useful to just be aware of what they are. These are sort of an internal scaffold that the cell has. They link into the cadherins on the cell surface, which are cell-cell junctions. They comprise things like the nuclear lamins, vimentins and keratins. In contrast, actins more associated with cell mobility, migration and cytokinesis. Vesicles can also travel along these actin fibres and you see a completely different pattern. These are made up of the protein actin, as the name suggests. But the type that we're going to focus on today are the microtubules. These are made up entirely of tubulin, and we're going to look at the different forms of tubulin that make up these microtubules. The microtubules are crucial for mitosis, so contraction of microtubules will cause chromatid separation, which we can see here. And this is the primary mechanism of action of antimitotic drugs. So where you've got microtubule contraction and dragging these two chromatids to separate microtubule organizing centers, then that is what the anti-microtubule drugs are doing, and we're going to focus on those in this session. So microtubules are absolutely crucial for chromatid separation, and what you can see here is you've got the centrosome with microtubules radiating out from that in prophase, so starting to get chromatin condensation and starting to see the chromosomes from forming. As we move on to prometaphase and metaphase, microtubules are binding to the chromosomes, so one microtubule on one side of the chromosome, so one chromatid, and another microtubule on the other chromatid. And then these are going to contract and pull one chromatid that way and one chromatid that way. And all the chromosomes are lined up on the mitotic spindle. So as we progress through to anaphase, you can see that we've got shortening of the kinetochore microtubules here, so they're bound to the chromatids. Those are shortening and those are pulling the chromatids this way and the others are going this way. At telophase we've got complete separation of the chromatids and we're starting to get the cell ready for cytokinesis, which is where the two cells separate. So there are several different types of microtubules. We're really going to focus on the kinetochore microtubules, which are these, which are involved in attaching to the chromatids and pulling them apart. The other microtubules are also central to mitosis. These astral microtubules often attach to the membrane and allow some sort of resistive force so that you can pull on this microtubule and this centrosome will remain in place. So these astral microtubules are important as well. These interpolar microtubules don't attach to the chromosomes themselves but seem to aid the alignment of the kinetochore microtubules. So they're all important, but it's the contraction of these that's important for halting M phase. So microtubules are made up entirely of tubulin protein, and this is heterodimers of alpha tubulin and beta tubulin, and both of these are GTP bound. And as shown in this diagram, you've got alpha tubulin, you've got beta tubulin. This is the GTP sites here and here, and these will stack head to head, so you've got alpha beta alpha beta alpha beta all the way up, and this is one protofilament, and then there are 13 protofilaments to make up a full microtubule. And these are stacked in such an arrangement that they are very slightly offset. So whereas you look down the centre of a microtubule, you see a lumen, which will contain something, but let's just call it a lumen from now on. you can see that the microtubules are arranged in such a way that they form a spiral around the outside of the microtubule, but importantly they arrange in very straight protofilaments. Now that's really important that those are straight when we come to look at the action of the drugs, because sometimes those drugs cause these to veer off at a slight angle, and that will destabilize that microtubule. Some of the interactions that we're going to look at involve the GTP hydrolysis. Now GTP is bound to alpha tubulin, GTP is bound to beta tubulin. When alpha and beta come together as a heterodimer, this alpha tubulin GTP is hydrolyzed to GDP, but the GTP on the beta tubulin is left intact. Now we then get incorporation of a tubulin heterodimer at the end of this protofilament, and this GTP bound beta tubulin, that presence of that GTP helps further addition of other tubulin heterodimers, creating what we call a tubulin GTP cap. So this GTP cap is crucial for extension of microtubules. If this GTP, which is on the beta tubulin, remains GTP bound, that encourages more and more tubulins to bind to this extended microtubule. However, if GTP hydrolysis occurs at this cap, we then tend to get disassembly of the microtubule. So as long as this GTP, as long as heterodimers are added in and the GTP doesn't immediately get hydrolyzed to GDP, we can keep adding more and we can extend that microtubule. Whereas if GTP hydrolysis occurs, then this tends to cause destabilization of the microtubule. So in this way the microtubules are incredibly dynamic, they can be extended and they can be disassembled very quickly. And this is just shown here. You've got an extending microtubule, tubulin dimers, heterodimers are being added, they're GTP bound. Now the GTP does get hydrolyzed, but as long as you're adding them in quicker than the GTP gets hydrolyzed, then we can continue to have extension. Whereas if that GTP is hydrolyzed en masse, the whole microtubule will then collapse. So as I mentioned earlier, these microtubules radiate from the spindle pole or centrosome or microtubule organizing center. And this microtubule organizing center comprises two centrioles at right angles to each other, and that comprises this centrosome. So from there we've got microtubules radiating out, attaching to the chromatids, and then being allowed to contract back and pull the chromatids towards that spindle pole. So that's the basics of the microtubules, tubulin, the microtubule organizing center covered. What we're now going to cover is how this can be drugged. And we've got lots of different classes of drugs that can bind tubulin, either alpha tubulin, beta tubulin, or various sites within that. So we're going to cover each of those in turn. So this image here shows alpha tubulin and beta tubulin. It shows the location of the main sites that are targets of drugs that target either alpha tubulin or beta tubulin. So the first one is the taxanes, and the taxane site is here on the outside of the beta tubulin. And what this does, it stabilizes GDP bound tubulin. So as we've just seen that GDP bound tubulin can depolymerize very effectively. If taxane is bound to that tubulin, then depolymerization cannot occur, and you lose your microtubule dynamics, and the cells then become stuck in metaphase, because metaphase is a time where the microtubule dynamics is absolutely crucial. We've then got the second main class, which is the vinca alkaloids, so vincristine, vinblastine are the main drugs that you're likely to come across, and they bind to the appropriately named vinca site. And the vinca site is on the top of beta tubulin. And the importance here is this vinca site, if the drug is bound there, an incoming alpha tubulin will try to sit on top of this, and if the vinca site is blocked with this drug, it will cause the next alpha beta tubulin heterodimer to fit into place at a slight angle, and it will create like a wedge effect, and this will form the protofilament to form at a slightly strange angle, not straight, and this will cause destabilization of the growing microtubule. And then the final main site of drugs, although there are lots more shown on here, is the colchicine site, which is a very well characterised site. And the colchicine site sits between the alpha and the beta tubulin. So when beta and alpha come together, if colchicine is present, they will either come together, or they will be inhibited in coming together, or come together less efficiently if the colchicine site is blocked. The other interesting site here is this one, the maytansine site. This is the drug, if anyone studied the drug Cellular, there was a tubulin inhibitor as part of that drug combination, and that binds to this particular site here. But I'm not going to cover that in detail. So this is just shown in a little bit more detail, showing the vinca site, which is present on top of the beta subunit. When vinca alkaloids are bound, that creates a little wedge, and that will push the growing protofilament off at an angle, and that will alter microtubule dynamics quite significantly. Whereas the colchicine site is between the alpha and the beta subunits, so that when colchicine is bound, this will form an abnormal alpha-beta subunit, which again will prevent extension, or it will prevent the formation of an alpha-beta in the first place. Since mitosis is a prerequisite for tumour growth, you would think that these drugs shouldn't have any resistance, but they do have resistance, and it comes from various different sources. Firstly, the taxanes. Now, if taxanes are present in the cell, they will always affect microtubule dynamics. The problem here is that the taxanes are very efficiently effluxed out of the cell. So paclitaxel, docetaxel, those are effluxed by P-glycoprotein, also known as multidrug resistance 1, also known as ATP binding cassette B1, so ABC B1. So if a cell overexpresses this, the taxanes are just pumped out of the cell and they never actually make it to their targets. So whenever a tumour is an MDR1 overexpressing, we know that the taxanes are not going to work. With the vinca alkaloids, only bind to certain isoforms of beta tubulin. So what a tumour can do is, if it naturally expresses one beta tubulin, and these beta tubulin isoforms are expressed differentially amongst different tissues. So if a particular tumour expresses a version of beta tubulin which vinca alkaloids bind to, then any cell that for whatever reason expresses a different isoform that vinca alkaloids don't bind to, they can just continue to express that isoform. So we know very efficiently in the lab, if we grow cells in the presence of vinca alkaloids, they will rapidly upregulate the beta 3 isoform to mediate resistance. Resistance of the colchicine type drugs is a combination of efflux, much like the taxanes, or beta tubulin mutations very close to the colchicine site. Now within this group, the colchicine type drugs, the main drug that has been used recently is the convertistatin drug, or CA4P. This is an anti-vascular drug, and it also has a different resistance mechanism that I'll come into at the end of this session. So for further reading on this topic of resistance to anti-tubulin chemotherapeutics, this is an excellent review which is well worth looking at, and this goes through both the changes in tubulin isoforms and tubulin mutations that mediate resistance to the various different tubulin binding drugs. So here you can see other mutations in tubulin beta 1 isoforms that cause resistance to paclitaxel. A lot of these are done in cell culture studies, and what these mean is, so for paclitaxel, which we've got here, F270V basically means a mutation that changes phenylalanine at amino acid 270 to a valine of the tubulin beta 1 coding sequence, and that will reduce paclitaxel drug binding. So these are additional mechanisms that are present that can cause resistance. Certainly in cell culture studies, if you follow up the references, you can find out whether these are in tumours or whether they're just in cell culture studies, but we know that that particular mutation will cause paclitaxel resistance. Similarly, there are other recurrent mutations that cause vinblastine and vincristine resistance, so D45Y, same idea. the 45 is the amino acid of the beta-1 tubulin, D and the Y are the amino acids. The first one is what the amino acid used to be before the mutation, and the Y is what the amino acid is after the mutation. You see that this one confers resistance to both vinblastine and colchicine. You can see other mutations also confer resistance to both drugs, and this is likely due to increased microtubule stability. And so here's just a reference that you might want to follow up, showing evidence of mutations in clinical samples in response to anti-tubulin inhibitors and emerging drug resistance. So here we're looking at about a third of samples with beta-1 tubulin mutations that are non-responsive to paclitaxel. So we can also see from this table that beta-tubulin isotype overexpression also can drive drug insensitivity, so either resistance or sensitivity. So we can see here we've got beta-2 isotype overexpression, and across a range of cancers this is causing docetaxel and vincristine resistance. And you get similar patterns for other tubulin isoform overexpression. So just an important point to remind you of is we've got clinical mutations of beta-1 tubulin, because that is what a lot of the drugs are targeted towards. And to get round that, cancers are either mutating beta-1 tubulin so that the drug no longer works, or the cancers are overexpressing other tubulins other than beta-1, because beta-1 is being inhibited, and therefore you can overexpress beta-2, particularly beta-3 as well. So the cells can get by using these other isoforms instead of beta-1, and the drugs do not bind anything like as well to these isoforms of tubulin than beta-1. So just by changing their expression profile, they can get round the activity of these drugs. So finally, looking at tubulin inhibitors, we've mentioned the compretostatins, which are part of the colchicine group, and the main drug that's been used recently is CA4P, and this targets the colchicine site of tubulin, but it doesn't primarily act upon the tumours, it acts upon the vascular endothelial cells of tumour blood vessels. So this drug is called a vascular disrupting agent, so we're not targeting the tumour cells directly, we're targeting the tumour's vasculature. So this just shows that blood vessels are comprised of endothelial cells which form the lumen of the blood vessel, then you get pericytes wrapping around the outside, and then for larger vessels you'll have vascular smooth muscle cells. So this is a lumen of a large vein, this is the lumen of a very small microvessel. So these drugs work primarily because vascular endothelial cells around tumours are very young, immature, the blood vessels are what we call immature. They're also very leaky and permeable. So this is the tumour that's been untreated. This has been treated with CA4P, and all of this region here is dead necrotic tissue because the vasculature has been shut down by CA4P. So by altering microtubule dynamics, the endothelial cells have lost their effectively watertight barrier, become incredibly leaky, and leaked out their contents into the tumour, and that's effectively caused venous stasis and shut down of the blood vessels, and therefore the tumour dies due to lack of oxygen and acute hypoxia. The problem is, and this is the main resistance mechanism here, is that these tumour blood vessels that are in this main tumour here are very susceptible, but the blood vessels surrounding the tumour are mature blood vessels which are not susceptible to the drug. And what you can see here at 24 hours is a very, very thin viable rim that is not dead, and by 72 hours that viable rim has started to grow again. So we get very rapid knockdown of the tumour, but then very rapid regrowth. So the mechanism of action here is not necessarily down to any mutation or change of expression of the target, it's just the fact that the tumour blood vessels that we are targeting, which is the immature vessels within the tumour, are very susceptible, but the normal vessels around the tumour, which are more established blood vessels, are not sensitive to CA4P, and they get recruited into the tumour, and then you end up with a tumour that no longer responds to CA4P. So if we go back to the resistance mechanisms of tubulin-targeted therapies, colchicine resistance can occur due to efflux or beta-tubulin mutation, but in the case of vascular disrupting agent compretostatin, the resistance mechanism is different. It's just down to the change in tumour vasculature compared to in tumours compared to normal tissues. So that summarises the role of anti-tubule binding agents in anti-cancer therapy.